Now, Farmer Jamie's not stupid. He knows what a test cross is, and he knows that this is what he's got to do in order to find out who the daddy is. So, is it necessary to call Maury, or can he figure this out on his own? We know that the sow was curly-tailed. Therefore, she had to be homozygous recessive. We're going to call that little t. Now, we have to figure out what the boar, a boar is an uncastrated male pig, what his genotype is by looking at the 10 piglets. Luckily, Farmer Jamie has a really smart girlfriend when it comes down to genetics and how to perform these crosses. So she addressed him with some important questions. First off, did all the piglets have curly tails? Did all the piglets have straight tails? Or was there a mixture between the two? This is going to help us determine what the dad's genotype might be. So question number 1, did all the piglets have curly tails? In order for all the piglets to have curly tails, knowing that curly is recessive, what must the dad's genotype be? It would have to be recessive, because remember, with recessive you have to have 2 alleles of it. There has to be 2 little t's. Well, the only way to get 2 little t's across the board, knowing that the mom's genotype is little t, homozygous recessive, the dad would have to be homozygous recessive as well. This will give you 100% of curly tails. Question number 2, did all the piglets have straight tails? With straight being dominant, we know we need a capital letter, right? We got to have something to show that dominant allele, that dominant trait. But do we need 1 of them or 2 of them? Well, in order for all of them to have a straight tail, we would need to have a homozygous dominance for the trait, meaning we have 2 big T's. If we had a big T, little t, what would end up happening would be in a 2-to-2 ratio, meaning that half of them would have straight tails and half of them would have curly. So we need them to all have that big T in it, which would make the dad homozygous dominant. And then the third question, of course, was there a mixture between the 2? If there was a mixture between the 2, meaning we have some curly, some straight, we would know that some would have to contain a dominant allele and some would have to have the some would have to be homozygous recessive, meaning they have 2 little t's. The only way to get some where we're going to have a big T and then some with 2 little t's, homozygous recessive compared to heterozygous, would be that we cross it with a heterozygous male. So he's big T, little t. This will give us 2 that are going to show the dominant trait, and this will give us 2 that are going to show the curly trait or the recessive trait. This does not mean that out of 4 animals, 2 of them are going to be straight and 2 of them are going to be curly. This clearly means that there's a 50% chance that they have a being straight or being curly. It's like with me and my brothers. There's 4 of us. It doesn't mean that I took square 1, Chip took square 2, Wesley square 3, and Wyatt square 4. This just means the percent of the chance that we had of receiving that square. So can we help Farmer Jamie by determining the piglet's dad's genotype? Absolutely, we can. Probability is the likelihood that a specific event will occur. It doesn't tell you exactly how many times it's going to occur, but it just gives you the chance that it could occur. It is expressed in words, as decimals, as percentages, or as fractions. Probabilities in genetic crosses do not show the definite outcome of a genetic cross. It's like I said with my brothers. It doesn't mean I'm going to get square 1 and Chip's going to get square 2 and Wesley square 3 and Wyatt square 4. It just shows the possibility that we could get that square. Meaning, again, if 2 heterozygous cross, there is a 25% that the offspring will be homozygous dominant, a 50% chance that it could be heterozygous, and a 25% chance that it could be homozygous recessive. It doesn't mean that 1 of them is going to be dominant, 1 recessive, and 2 of them heterozygous. They do show the possible outcomes. Probability is the number of ways a specific event can occur divided by the total number of possible outcomes. So some practice problems. Probability is something you do in math You've been doing math for quite a while. But here's just some basic problems. What is the probability of flipping heads on a quarter? Well, we have two sides on a quarter. So how many of those sides are heads? One. 1 out of 2. You got a 50% chance of getting heads. Doesn't mean every time if I flip the quarter twice, one of them is going to be heads. It just means I have a 50% chance of getting heads. What is the probability of drawing an ace from a deck of cards? Well, in order to solve this, you have to know how many cards are in a deck. There's 52 cards in a deck. How many suits are in a deck? There's 4 suits. Well, out of those suits, how many of those are aces? Well, there's 4 aces, so 4 out of 52. Yes, you can simplify that, but for the sake, it's 4 out of 52, or 1 out of 13. What is the probability of rolling a 3 on a dice? Well, how many sides are on a dice? 6. And how many of those sides has the number 3? 1. So you have a 1 out of 6 chance of rolling a 3 on a dice. Dihybrid crosses are something that, when it comes down to genetics, a lot of people have trouble with. When you get into your upper genetics, you actually have more than a dihybrid cross. This is just the very basic in your college level genetics classes. So for a dihybrid cross, for your sake, is the most extreme one that you'll hit, and it has 16 squares. Mendel's dihybrid crosses led to a second law, the law of independent assortment. This law again states that allele pairs separate independently of each other during meiosis, meaning having freckles does not mean you're going to be short. A dihybrid cross is a cross that shows the possible offspring for 2 traits. This means with our alleles, we're going to have 4 of them. It's going to be like big R, little t, something like that. And again, it has 16 squares. We're going to work through a practice problem, and of course it's going to involve pigs, and so you can get a better understanding. Dihybrid crosses as well have something to do with math in a way, and the way that you're going to relate them to the FOIL method. For our example, we're going to use a pink pig and a blue pig. These pigs are going to either have spots or they're not going to have spots. So for the pink pig, when I do crosses, I like to use letters that are obvious to tell the difference between a capital and a lowercase. For example, P's are really hard to tell because sometimes you can't tell if you wrote the lowercase or if you wrote a capital. Same for S's, same for Z's, V's, you know the different letters. So I use Q's and B's. Q's are obvious to tell whether you have a capital or lowercase, same for B's. So for the pink pig, it's a capital Q, meaning it's dominant. And for the blue pig, it's a lowercase q, meaning it's recessive. Same for spots. For spots, it's going to be a capital B for dominant, and no spots is going to be a lowercase b for recessive. So the cross that we're going to perform today is going to be a heterozygous pink spotted pig crossed with a heterozygous pink spotted pig. And you can see the images that I have shown on the screen just to give you a visual as to what we're actually doing. The very first thing we need to do first off is to draw out your 16 squares. It's going to take quite a bit of paper to do this. So you have your 16 squares, which I have on my PowerPoint as well. But what goes where? So when you do a monohybrid cross, only one letter goes on top, and this is where a lot of people get confused. They'd like to write across the top, big Q, then the next one, little q, then a big B, then a little b. This is not correct. If you do this, you will get it wrong. What you have to do is you have to use the FOIL method. Again, you do not write one letter at the top and the side of each box. On the top and the side of each box must be 2 letters. There's got to be 2 for each square going across the top and down the side. So in order to get the 2 across the top and the 2 down the side, we have to do the FOIL method. The FOIL method is something you do in math. So the FOIL, you got to do the first ones first. So you do the first of each letter, the big Q, the big B. As you can see, that goes up the top. For this example, since we're crossing 2 of the same things, if it's across the top, it's going down the side. This doesn't mean it's the same for every example, but in this case it is. So the next one is you do the outside ones. Well, the outside ones in this case are going to be your big Q and your little b. They're on the outside. Then you do the inner ones. So that's your little q and your little b, or your big B, I'm sorry, little q, big B. And then you do the last. Well, the last in each set of these traits is going to be your little q and your little b. You can see I have this set up. Then we do the same thing like we did in a monohybrid cross. You pull down from the top and over from the side. Always make sure you write the same letters next to each other. So don't, for example, on the first square do big Q, big B, big Q, big B. No, you want to stick your letters together for the same trait. So do big Q, big B, big B. And you're going to go through the whole square doing the same exact thing. Really easy, as long as you remember to pull from the top and pull from the side. So just doing the square, the dihybrid cross in this example, that's the easy part. The harder part sometimes comes down to figuring out what you actually just did. So in this example, I've drawn out exactly what you have crossed. So what type of pigs did we end up with? Well, we ended up with 9 pigs having a big Q, so a dominant Q, meaning it's pink, and a dominant B, which stood for spotted. So we had 9 pigs that have this. You can see that they run all down the first row and the first column. And then we have 2 random ones in there as well. And then you have 3 of them that were dominant for pink, but they had 2 little b's. So they were a big Q, little b. So they're not spotted. We had 3 of them that were recessive for blue. They had 2 little Q's, and they had at least 1 big B. So they were blue and spotted. And then we only had 1 that was a little Q, little b, meaning it was recessive for both traits. So it was blue and not spotted. This type of ratio is one that you should memorize. When you cross 2 heterozygous things in a dihybrid cross, you will always end up with a 9-to-3 to 3-to-1. Remember, these have to equal up to 16 to know that you're correct. And yes, it does. So if you cross 2 heterozygous in a dihybrid cross and you do not end up with a ratio of a 9-to-3 to 3-to-1, you have done something wrong. This is something that you can easily go back and check yourself on. So for this slide, I pretty much described it earlier. Again, you're going to have a 9-to-3 to 3-to-1 ratio, meaning we have 9 showing both traits dominant, big Q, big B. 3 showing one of the traits dominant and the other recessive, big Q, little b. Again, 3 showing one of the traits dominant, the other recessive, in this case, little q, big B. And we have one that has homozygous recessive for both traits. When you add up your numbers, again, it has to equal 16. If you do not end up with a 9-to-3 to 3-to-1 with a heterozygous dihybrid cross, I know that's a lot of big words, you have done something wrong and you need to go back and check your work.